Welcome to this NanoSIMS webinar. Today we will start with basic isotopic notions, followed by some NanoSIMS applications in the field of lipid studies when coupled with an isotopic labeling strategy. Refer to our other webinars or support for instrumental aspects. My name is François Auréat. I am the NanoSIMS product manager at Chemica France. So do not hesitate to contact me. My email is on this page. First, there are the chemical elements listed in the periodic table of elements. The table is organized from left to right and top to bottom by increasing weight or proton number inside the atom nucleus. The number of protons is always equal to the number of electrons. It's called the atomic number. The rows of the table indicate the growing number of electron shells, and the columns indicate the same number of electrons on some electron subshell. What is the key of our topic of isotopic labeling is that the chemical reactivity is governed by the electron configuration, not the mass of the atom. Finally, the colors give the families linked to the physical characteristics or reactivities like alkalis, metals, etc. So isotopes have different masses, and we will be able to differentiate them with a mass spectrometer. They have the same chemical reactivity, so we'll be able to build experiments using this characteristic. Let's see, for example, selenium. On the right side, I can see number 34. This is the atomic number, the proton number. And on the left, I can see the atomic mass, which is not an integer. And why? Because it's a mixture. The natural selenium is a mixture of different isotopes with different weights. Stable isotopes have fixed natural ratio on Earth. And so the strategy with the NanoSIMS is this one. Molecules artificially enriched or labeled with one or several rare isotopes with a low background can be introduced in a biological system through the food, the substrate, medium, by perfusion, or through already enriched cells. And their distribution can be mapped and followed over time with the NanoSIMS through the measurements of the local isotopic enrichment down to a local resolution of 50 nanometers. Let's continue on the selenium example and use the article of Tang et al. In Surface and Interface Analysis in 2014. So selenium is an essential micronutrient in humans and animals. Biological activity mainly exerted through selenoproteins that contain selenocysteine, the 21st amino acid. And selenoprotein synthesis involves selenide, precursor of selenophosphate used to synthesize sec-RNA transporter RNA, a prerequisite step for selenium to be incorporated into selenoproteins. Here we see different synthesis pathways for selenoprotein used by humans and animals. Now in this mass spectrum, we see again the several isotopes of natural selenium. So, in this experiment, human aortic endothelial cells were incubated during 4 days with different monoisotopic selenium precursor compounds. It was then possible to map their internalization and compartmental localization through their specific isotopic ratio. First, a few words on the various possibilities of sample preparation So these aortic cells were grown on gold-coated silicon chips. They were frozen and freeze-dried, and the dried cells were bombarded from the top with the NanoSIMS ion beam in order to sputter through. We see such CN- image, so mostly the protein distribution. And now, so we see the results of the study of this article. We see the 3 selenium isotope mappings from the 3 different labeled selenium precursors, and their distribution is evidently different. So different isotopes of one element have different distribution. They can be used to study metabolic pathways. So this example illustrates the NanoSIMS capability to make use of an experiment based on multiple isotopic labeling. Coming back to sample prep, note that this type of top-down analysis can take time. So an alternate preparation is resin embedding and microtome section, very similar to TEM analysis. And it's faster. On the left side, we see the cells that have been detached from the silicon substrate using trypsin. That gives them a round aspect. On the right side, a different strategy of preparation, giving them a more natural shape. So, in summary, isotopes have same chemical reactivity, but different mass over charge ratio. They are undiscernible by cells and tissues. So it's possible to build experiments based on the supply of one or several isotopically labeled compounds to living cells, tissues, plants, or animals. So it's wiser to use a low intensity isotope with a low background. The NanoSIMS will allow mapping the distribution of and fluxes of such isotopes with 50 nanometer resolution. And any isotope can be used, but stable isotopes like deuterium are advantageous over radioactive like tritium because they have not to deal with safety issues. Let's start with application in lipids. And we will use this scientific article by Q&A in Cell Metabolism 2018 on intravascular lipolysis and lipid movement across capillaries and into cardiomyocytes. So in short, they use the NanoSIMS to visualize the movement of triglyceride, which lipoprotein-derived lipids in the heart. So the triglyceride-rich lipoprotein enriched in deuterium are injected intravenously to mice. After a given time, the mouse is sacrificed, fixed. The tissues are prepared as thin section, and NanoSIMS imaging is performed to visualize the distribution of deuterium-labeled lipids in the heart. So CN- images are used for the histology, showing the cardiomyocyte and capillary endothelial cells in the heart, while the deuterium over hydrogen ratio images reveal the deuterium-labeled lipids from the TRL into the cardiomyocytes. First, we see two representative images of heart tissues, one above the other, 30 seconds only after injection of TRLs. On the left, one can see capillary inside red square in the black and white CN image, surrounded by cardiomyocytes. And on the right side images, one see in red color moles, the areas enriched in deuterium from the injected lipids. Some lipid droplets are marginated along the capillary endothelial cells, marked with white arrows. But the striking point is to see already many deuterium-rich lipid droplets already inside cardiomyocytes after only 30 seconds. 2 minutes after the deuterium-labeled lipid injection, and of course it's from a different mouse, the number of marginated lipid droplets has reduced. And 30 minutes after the injection, all labeled lipids are found inside the cardiomyocytes. We can zoom in the red squares of above images to have a finer view of the movement of lipids in the tissue. Here we can measure the deuterium over hydrogen ratio is up to 5-6 times higher than in the natural ratio. So the two main outputs here: first, intravascular lipolysis can be visualized by NanoSIMS, and second, TRL-derived lipids move across endothelial cells and into cardiomyocytes, mitochondria, and lipid droplets within seconds. We now see 3 locations of capillaries surrounded by endothelial cells and cardiomyocytes all around. These images are very shortly after TRL lipid injection, 30 seconds or 2 minutes. We can observe in red marginated lipid droplets. Field of view is 10 by 10 micron. The NanoSIMS resolution is 50-100 nanometer. From these images, it is then possible to extract quantitative values from small spot area moving from the droplet to the lumen labeled L, or along the endothelial cells labeled E1 or E2, or into the cardiomyocyte labeled C. So these sorts of line scans are displayed for the 3 examples for deuterium and CN. On the deuterium over hydrogen signal, one can clearly see a trend of decrease when moving from the droplet, evidencing that the lipoprotein-derived lipids do not accumulate in endothelial cells as it has been suggested in some previous works. So NanoSIMS imaging opened the door to examining the functional relevance of specific proteins in the movement of TRL-derived lipids into and across capillaries. And here, CD36 protein, a putative fatty acid transporter, is expressed at high levels in hard capillary endothelial cells, and it has been hypothesized to play a role in the movement of fatty acids into cardiomyocytes. So this will be tested in this part of the experiment. Wild type and CD36 deficient mice were injected intravenously with equivalent amount of deuterium TRLs, and we display the images recorded 1.5 and 2 minutes after the injection. So above the wild type and below the 2 rows of deficient mice. And one can see that we see less droplets in myocytes of deficient mice below. But if you extract the data quantitatively from the images, you see that the quantitative value of deuterium enrichment are very similar for both mouse lines. Also, it was observed that the fewer lipid droplets present in the deficient mice, cardiomyocytes, contain clearly a lower percentage of deuterium. This aspect is less understood. So, a conclusion is that the TRL-derived lipids move across endothelial cells and enter cardiomyocytes in both wild-type and deficient mice. So, CD36 protein deficiency does not prevent entry of TRL-derived lipids into cardiomyocytes. So some outputs of these studies. In terms of results, we have seen that lipoprotein-derived lipids enter cardiomyocytes within seconds, very fast. They do not accumulate in endothelial cells. You could see this with the resolution of the NanoSIMS. And CD36 protein deficiency does not prevent entry of TRL-derived lipids into cardiomyocytes. Now, in terms of NanoSIMS methodology, we can see that intravascular lipolysis can be visualized by NanoSIMS. Deuterium label precursor can be used to follow the incorporation and localization of lipids with typically 50 nanometer resolution in cesium. A CN- image gives an excellent anatomical recognition without any staining. So sometimes, also for histology, in other cases, we follow sulfur or oxygen or phosphorus and even secondary electron images. And fast response experiments are accessible. Here, for example, 30 seconds, but know that one animal is required per time point in this study. If it would be cell, it would be a certain number of cells sampled at a certain time. Anyway, it is not a high throughput technique. So we will now use a part of this article by Stenhauser et al. In Nature, 2012. We will look at lipid turnover. The Drosophila fly provides a model system in which to study the genetics of lipid metabolism in vivo. The speed of reproduction is high, but the animal is very small for conventional mass spectrometry. So the NanoSIMS is used here to measure an image, the incorporation and turnover of diet-derived C13 labeled palmipate within cells. So there will be two types of experiments. C13 palmipate-rich diet pulses, variable duration, to study the storage evolution, and also fixed C13 palmipate pulse duration. Here it will be 24 hours, followed by chase without C13 palmipate of different duration to measure the decrease. After 24 hours of feeding carbon-13 enriched food, we see on the left side a CN image in black and white and a carbon-13 ratio image of fat body cells, labeled FP. The lipid droplets of fat body cells appear in black in the nitrogen map, and they appear in light blue in the C13 image, revealing an enrichment above the natural ratio that is in dark blue. Note that the lateral resolution of the NanoSIMS in this experiment is roughly 50 to 100 nanometer. But it was a surprise to see that the carbon-13 palmitate was stored in some lipid droplets in the gut at a higher level compared to the expected location of fat body cells. So Drosophila, anterior midgut enterocytes, accumulate more C13 from 24 hours exposure to dietary C13 palmipate than adipocytes. The experiment was repeated, varying the pulse duration, each point coming from a different slide, of course. So we see that a faster and higher palmipate accumulation in the gut droplets compared to the fat body cells. At this point of this article, we can stop and make a pause to highlight three instrumental aspects. So first, the notion of ratioing two isotope images recorded in parallel from the same volume cancels most potential sputtering, ionization, or topographical artifacts. Also, we have an internal standard, the natural ratio of the embedding resin, that cancels most of the potential instrumental fractionation artifacts. So, reliable method. Second, on the left side, you see color images. They are displayed in HSI color scale, hue, saturation, intensity. So, one pixel with a given isotopic ratio acquired with a high intensity, for example, 50 counts on the C13 and 5,000 counts on C12, will appear with the same color as another pixel with 2 counts on the C13 and 20 counts on the C12. But they will have the same hue, but as the signal to noise is much lower, they will appear in this display mode much darker, so less visible. So we see only mostly the statistically significant pixels. Third aspect, how do we get these quantitative data? We draw manually or with some software a region of interest in the image, and we sum all pixel intensity from both isotopes. So we get a better isotopic ratio signal to noise. So let's come back to the article and the second part, the chasing part. After accumulation, the images are fed natural diet for variable times now. We see the decrease of carbon-13 palmitate after 4-hour chase. So this is clearly visible in the color scale for the isotopic ratio. So there is a rapid lipid turnover in the anterior midgut enterocytes after larval dietary C13 palmitate pulse chase. And if we can record on different animals the C13 tracer dilution in the lipid droplets of midgut enterocytes, and it fits very well an exponential decay. So let's see some outputs of this publication. Concerning the results, the palmitate label is incorporated into enterocyte lipid droplets with a subcellular pattern similar to that's seen with a neutral lipid dye, so validating methodology. The C13 palmitate pulses of varying periods indicate that the rate of carbon-13 label incorporation into lipid droplets is greater in anterior midgut enterocytes than in the adipocyte of the fat body, the major lipid storage depot in Drosophila. And a pulse chase experiment with dietary C13 palmitate also demonstrate a reproducible exponential decay of the carbon-13 signal within enterocyte lipid droplets and with an half-life of approximately 9 hours. And concerning the methodology, we have seen that it's possible to measure lipid turnover from small areas with 50 nanometers lateral resolution. Carbon-13 label is perfectly usable, although, or despite the presence of a mass interference, C12H, very near, but no problem for the NanoSIMS with its high mass resolving power to separate this. The sensitivity is good, throughput is good. And we have seen the method of ratioing the isotope images. They are recorded in parallel, and we have an internal reference from the embedding medium. So we remove most methodology artifact, and the HSI display of ratio images improves the visibility of statistically meaningful pixels. This next example in lipid field will be taken from the article published by Andrew Bailey in Cell in 2015. So, in very short, lipid droplets can act as antioxidant organelles that protect Drosophila neural stem cells from hypoxia-triggered reactive oxygen species, ROS. And these lipid droplets sequester polyunsaturated fatty acids, PUFAs, away from plasma membranes, thereby shielding these lipids from harmful peroxidation. The NanoSIMS measurements are only a part of the study. So the question to be answered by NanoSIMS is, what are the constituent origins of these lipid droplets induced by hypoxia? This answer is solved by raising larvae on different diets containing different labeled molecules. So the researchers recorded images of glial lipid droplets induced by hypoxia, larvae raised on diets containing whether C13 labeled glucose or C13 acetate, both major carbon sources of lipogenesis. So you can see after hypoxia the appearance of small lipid droplets of sub-micron size to 1-2 micron synthesized during the lipogenesis. But one can also see that the similar droplets appear also when feeding an essential, so non-synthesized PUFA. Hence, coupling stable isotope labeling experiment with NanoSIMS mapping allowed answering clearly the question. So, the outputs, first on the results, on the publication, the oxidative stress stimulates lipid droplets biosynthesis in a neural stem cell niche. Lipid droplets protect niche and neural stem cells from damaging PUFA peroxidation. And PUFA are less vulnerable to peroxidation in lipid droplets than in cell membranes. And in terms of NanoSIMS methodology, coupling the NanoSIMS with experiments using stable isotope labeled diets can elucidate the metabolic origin of lipid droplets. Here, carbon-13 was used. The technique is applicable with the lateral resolution down to 50 nanometers. Note that smaller objects can be detected, but will appear with the size of the ion beam. The ratio images are displayed in HSI color scale, validating their statistical significance. We will continue and use this paper of Jessica Fritz in PNAS in 2013. So sphingolipids play important roles in plasma membrane structure and cell signaling. However, their lateral distribution in the plasma membrane is poorly understood. This study is focused on the quantitative analysis of the sphingolipid organization on the entire dorsal surface of intact cells. The lateral distribution of metabolically labeled nitrogen-15 sphingolipids in the plasma membrane is obtained by imaging the distribution of nitrogen-15 enriched ion, and the distribution of carbon-13 labeled fatty acid is mapped in parallel for checking membrane integrity. So, after having seen sections of cells, we will now see surface of cells analyzed with the NanoSIMS. And we will illustrate here the possibility to use simultaneously multiple isotopic labels and map their distribution in parallel. So the method, we see a general structure of sphingolipid with two parts. So clone 15 fibroblast cells were metabolically labeled using fully carbon-13 labeled fatty acids and the nitrogen 15 sphingolipid precursors, so sphingosine and sphinganine, such that the resulting sphingolipids reached a steady state distribution in the plasma membrane without nitrogen 15 incorporation into proteins. The uniformly carbon-13 labeled fatty acids varied in chain length and degree of saturation to promote their incorporation in all sphingolipid and glycerolipid species, presumably producing an even distribution of carbon-13 lipid in the cell membrane. So this global labeling of most lipid with carbon-13 was meant to evaluate the plasma membrane integrity and possible artifacts. We show here a mosaic of secondary electron images collected in the NanoSIMS in parallel with the ion images. SE images greatly help in observing the morphology of the cell, here fixed and air-dried on a flat silicon substrate. Now, on the right side, we display the carbon-13 enrichment, which is the carbon-13 over carbon-12 ratio normalized by the natural isotopic ratio. We do not observe significant localized enrichment, but rather an homogeneous signal indicating that the plasma membrane has been well kept intact. Finally, we display the nitrogen 15 enrichment, revealing small domains in the membrane, strongly enriched in nitrogen 15 sphingolipids of up to 30 times the natural ratio. Now, in terms of instrumentation and method, 5 images have been recorded in parallel. So secondary electron, carbon-12, carbon-13, CN14, CN15. CN ions are used to map nitrogen in carbon-containing sample, giving the best sensitivity. So the challenge of carbon-13 mapping lies in the huge carbon-12H peak near the small carbon-13 peak. Mapping nitrogen 15 is even more challenging, as carbon-13 nitrogen 14 is even closer to carbon-12 nitrogen 15. And so the strength or the uniqueness of the NanoSIMS is to ensure high mass resolving power to separate the peaks, while keeping high sensitivity and 50 nanometer spatial resolution. Note the sharp mass peak side and the high dynamic range shown in logarithmic scale. We present here two other cells out of the many cells that have been analyzed for this publication, showing similar results. A control cell show evident lack of enrichment. And we have already mentioned or seen the interest of using ratio images, allowing to cancel topographical artifacts, another possible artifact. And one thing to mention is that it is very useful, again, to have this internal standard and validity given by the embedding resin. In a following step, a detailed statistical analysis of the size of these small sphingolipid domains has been performed, revealing a mean domain diameter of 200 nanometer in the plasma membrane. So some outputs of this study. First, in terms of result, micrometer scale sphingolipid patches consisting of numerous nitrogen 15 sphingolipid domains of mean diameter 200 nanometer are always present in the plasma membrane. The depletion of the cellular cholesterol did not eliminate the sphingolipid domains, but reduced their abundance and long-range organization in the plasma membrane. In contrast, disruption of the cytoskeleton eliminated the sphingolipid domains. So these results indicate that these sphingolipid assemblages are not lipid raft and are instead a distinctly different type of sphingolipid-enriched plasma membrane domain that depend upon corticolectin. In terms of NanoSIMS methodology, we have seen the possibility to use nitrogen 15 label, keeping extreme sensitivity and spatial resolution despite the high mass resolving power required to resolve mass interference or potential mass interference from C13. We have seen that a multiple labeling strategy is possible with multi-collection. Here it was carbon and nitrogen in parallel for isotopes, but other publications have shown, for example, deuterium, carbon-13, nitrogen 15, and O18. Since a high depth resolution allows thin membrane analysis from top view mapping, so few nanometers. And we have seen also secondary electron images induced by the primary ions are very useful for topographical images and recognition, and mapping of large areas up to 100 micron, if necessary, is performed in mosaic mode. So, general conclusion. The NanoSIMS 50L is a unique ion microprobe combining simultaneously 50 nanometer lateral resolution, parallel detection of 7 masses, and high mass resolving power. The strategy of using one or several stable isotope labelings opens new fields of study in life science generally and in lipids studies in particular. The NanoSIMS offers a powerful and quantitative technique for mapping and studying lipid origin, distribution, fluxes, kinetics, and metabolism at 15 nanometer lateral resolution. So most scientific publications using the NanoSIMS are compiled on the Chemica website in an Excel spreadsheet facilitating the search. You can download it in the NanoSIMS page. Feel free to contact me by email or through your local agent for questions or information. Thank you for your attention.